Yeah, thank you again for the introduction and the invitation. I'm excited to be able to share some of the work that we've been doing in my lab over the last few years. My lab is in mechanical and aerospace engineering at Ohio State University. You see a picture of the folks in my lab. Actually, not all of those people in our lab. We're very collaborative and interdisciplinary. So we have a mixture of mechanical engineers, biophysicists, chemical engineers, and some biomedical engineers. And some of the folks there, I should point out, are from collaborators' labs, but work very closely with our lab. And so, yeah, we focus in DNA nanotechnology, and kind of a driving direction for our lab is trying to engineer these DNA devices to physically probe biomolecules, but also moving towards doing that in situ, meaning maybe in live cells. And so those are kind of moving directions in parallel, but we hope to integrate those two in the not too distant future. Um, so just by quick conflict of interest disclosure, I'm co-founder. We spun a startup company out of my lab a few years ago that has some peripheral interest in some of the stuff I'll talk about today. And we do have a provisional patent on this, what we call free energy spectroscopy, that I'll talk about later in the talk. So this is a graphic that I like to use to introduce what we do in the lab. Our lab is very good at this advanced design of nanomechanical devices. If you're familiar with DNA nanotechnology or DNA origami, the particular approach that we use, you may be aware that it's quite powerful in terms of the complex nanoscale geometries that you can achieve as an example structure here. In our lab, we also think a lot about not just the geometric design, but how do we design in some specific mechanical or dynamic properties, often in the interest of having, some biophysical probes. And we are often interested in interfacing with biological systems, either cells or molecules. And so we're often functionalizing with things like antibodies, or maybe putting fluorophores on for readouts. And then the directions we take these devices in is one kind of still a little bit more fundamental direction of nanorobotic devices and materials, where we're trying to leverage these nanomechanical devices to, say, manipulate or assemble other materials or particles. A lot of our work, and this is what I'll focus on today, is in trying to make nanoscale measurement tools for biological systems. And we do have a couple projects that are really geared more towards direct biomedical applications, such as non-viral gene delivery, something we're working towards now. Because of these two directions, again, I'll focus more on the biophysical measurement tools, but because of these two directions, we kind of have a broader interest on how do we deliver nano devices to these specific locations in a cell or tumor microenvironment. And so I'm highlighting some of these locations of interest here, but in the later part of the talk, I'll show you how we're specifically working towards delivering devices directly to live cell nuclei. So I'll start with giving a little bit of a background on just the basics of DNA origami in case folks are not familiar. I will very briefly show, just more of an aside highlight. We've developed some protocols to implement that in classrooms. So in case people are interested, I'd just like to share that. And then I'll spend most of the time talking about how we're designing devices to probe biomolecules, in particular nucleosomes and chromatin is our current focus. And then at the end, I'll spend a few minutes telling you about how we're designing these devices to hopefully get them into cell nuclei, and over the longer term, maybe perform some of these kinds of measurements or manipulations inside of live cells. So if you're not familiar with DNA nanotechnology or structural DNA nanotechnology in particular, I often like to start with, well, why are we building structures and devices and materials out of DNA in the first place? Biologically, it's not really thought of as a structural polymer, but it turns out it has some pretty nice properties that make it useful for programming self-assembly of structures and devices. And first and foremost, it's that the rules that govern the self-assembly of DNA are quite simple. And we have AT and GC base pairing, and basically that's what governs the pairing and binding of these two strands into the DNA double helix. Of course, biologically, it's very important. So it's been studied for many, many decades, and that's led to a very well-known and well-defined structure and mechanical properties. And it turns out DNA is flexible, but I guess the important part, which you'll get a sense of later in the talk, is that we can actually tune the mechanical properties of components. For example, we can use single-stranded DNA if we want something quite flexible, or we can bundle together several helices of double-stranded DNA if we want a component that's quite stiff. And so that allows us to kind of traverse this range of component stiffness and build up these nanomechanical devices. And so coming back quickly to this idea that the rules that govern self-assembly are quite simple, that really allows us to do what we call program self-assembly, where if we have a template strand of known sequence, we can pretty easily design another strand that should bind to it, just following, again, these ATGC base pairing rules. And if we mix these two together in the right solution conditions, we should get this nice DNA double helix. And of course, we're interested in building things a little bit more complex than just a double helix. And so we use what's called this scaffolded DNA origami approach that was developed 20 years ago now by Paul Rodemund, a research scientist at the time at Caltech. And in this approach, we, just like we did on the previous slide, we design sequences that are complementary, but in this case, to different regions of the scaffold. So for example, the blue to blue, red to red, and green to green. And then we actually stitch those sequences that should bind to 3 distant regions of the scaffold strand together continuously on one strand that we refer to as a staple strand. And if we mix those together, that scaffold has to fold up into some more compact shape in order to continuously bind to that one strand. And so this quite literally staples different regions of the scaffold together. And so this was first demonstrated again about 20 years ago with some of these very impressive nanoscale shapes. And if we scale this out to each scaffold is typically folded by maybe 150 or 200 staples, you can see what that looks like for a more complete folding reaction. We mix the scaffold and staples. You start pinching the scaffold together to ultimately form some nanoscale architecture. This is a typical kind of blueprint view that you see the scaffold and staple routing, but really that's consisting of a bunch of double-stranded DNA helices in parallel that are connected together at these places where either the scaffold or staple strands cross over from one helix to the neighboring helix. And these are often depicted as pretty stiff, solid structures, but there is a lot of local flexibility. This is just a coarse-grained molecular dynamics simulation of this particular design. You see it maintains the overall geometry, but there is a lot of local flexibility, which can be important to account for in some cases. And so I won't talk much about the fabrication, but I just wanted to give you a sense for the fabrication, right? We typically mix these, again, in certain ion conditions. There's a thermally controlled annealing process that controls a self-assembly reaction. This is carried out in typically a thermocycler or PCR machine. And this, each fabrication can make a few micrograms, and the time it takes is, anywhere from about 10 hours up to a couple days. This works well, especially for research scale. Our lab and other labs have developed methods to scale and simpler methods, especially if we're thinking about clinical applications. It's helpful to scale to, say, hundreds or thousands of micrograms, milligram scale. And just as a brief aside, we've also developed some pretty cheap, simple, easy protocols to actually do this in a classroom with pretty low-cost equipment. I won't talk much about this, but I like to share this in case anyone has interest to try this out. I'm more than happy the papers are there, everything's there that you need, but I'm more than happy to share protocols and give some advice or feedback on how to implement this in a classroom. So just coming back to what you can do with these structures, right? There's a huge geometric design space. None of this work is from our lab, but just to highlight the diversity of geometries that you can make. One of the things our lab has been very interested in is how do we make these devices dynamic, right? So how do we make them reconfigurable? And here I'm just showing highlights of this kind of nanorobotics direction. These are 2 examples that we published a little while ago now, but this is a 3D linkage that can open and close between an open frame and this closed bundle. And this is actually a paper origami inspired design, this water bomb base that can reconfigure into 6 different states, conformational states. More recently, we've been really interested in scaling this up, right? How do we scale this up to get larger displacements and property changes? And how do we do more complex functions like signal transmission? And again, this is not more a robotics direction that I won't talk about today, but of course, I'm happy to talk about offline. But today I'll tell you more about how are we engineering these devices to physically interrogate biomolecular complexes. And this has been a pretty active direction for the field. I'm just highlighting some different examples here. Some of the earliest work, including ours on the top left, focused actually, interestingly enough, multiple studies focusing on studying nucleosome unwrapping or stacking interactions. And more recently, folks have implemented these types of clamp devices, as you see on the bottom, to study, say, DNA-protein or protein-protein interactions. This is a cool example from Chen-Chen Lin's lab that was published last year, where they actually apply different amounts of force to this tailwind subdomain and use this in a pull-down assay to see how force applied on this domain affects the interactions of this domain. Of course, I'll focus more on our work. And this has been a long-standing collaboration with Michael Poyer and Ralph Boonshoe, who are both in the physics department here at Ohio State University. First I want to introduce, we've been interested in using these devices to study nucleosomes and chromatin subunits. Nucleosomes are the fundamental packing unit of genomic DNA in eukaryotic cells. You have this DNA that gets wrapped about 1.5 times around this histone protein core. And these nucleosomes appear kind of as these beads on a string in the genome. And then those nucleosomes fold up into different levels of higher order structure. We are particularly interested in these kind of lower levels of the hierarchy where you have a few nucleosomes that wrap up into higher order structures. There are many other biophysical methods to study these, including things like single molecule FRET or force spectroscopy, and this has been, of course, very active and useful directions of research. I won't go into this much in the interest of time, but I hope to show you that we have these new approaches and new tools that are synergistic and in some cases have some clear advantages relative to these other methods. And so our general idea was to develop these nanocalipers where we can incorporate a biomolecule, right? This biomolecule that we've incorporated, which started with single nucleosomes, and now you'll see we're moving to multinucleosome subunits of chromatin. And we wanted to have this nanocaliper that could measure nanoscale distances, measure conformational changes, apply forces, and maybe measure the mechanical response of these molecular complexes. And eventually, right, I'll show you a little bit more towards that in the later part of the last part of the talk, move this towards studying chromatin in live cells. But we started, of course, with how do we design this tool? This is actually maybe the third or fourth iteration of these caliper tools that we're still currently working with that we've published 5 years ago now. This is the work of 2 PhD students from my lab that graduated a little while ago now. But we have these 2 stiff arms, DNA origami of DNA origami, that are connected together by these flexible single-stranded connections. And it's a little bit hard to see, but you see these short black connections are actually 2 nucleotides of single-stranded DNA. And those actually define the axis of rotation. And then we have these longer, more flexible single-stranded DNA that are 70 bases long. And those really are important in influencing the mechanical properties of the opening and closing of this caliper device. And so the way we characterize these is we fold them, right? We measure, we image them by transmission electron microscopy. This is a 50 nanometer scale bar here. And then we basically collect and quantify the angle distributions and then use, assuming a Boltzmann distribution, we can turn that into a free energy landscape here. And the basic idea here is that if we change the mechanical properties of these linkers by base pairing them in different ways, we can alter the mechanical properties and shift around this free energy landscape, ultimately allowing us to have calipers with different mechanical properties that can apply different levels of force to the biomolecules that we incorporate between the arms. And so kind of by intuition-driven brute force, although now we're trying to understand how to do this more systematically currently, but we have been able to develop these caliper tools where we can really shift around the free energy landscape. The red here is the single-stranded case, and then we can either push it more closed to smaller angles or more open to larger angles. And again, that allows us to have these kind of set of caliper tools that can apply different amounts of force ultimately to a sample that's incorporated between the arms somewhere. And the sample we've been working with lately is this tetranucleosome subunit of chromatin. These nucleosomes in cells have been observed to form these clusters or clutches, and other work has identified that these tetranucleosome subunits or units of 4 nucleosomes are an important regulatory unit in cells. Right? And we're particularly interested in studying the structural dynamics of those tetranucleosomes, which basically consist of stacking and unstacking of these nucleosomes and then wrapping and unwrapping of the DNA around those nucleosomes. And so this has been a long effort. This is the work I'll present now is the work of Calvin, who's a PhD student in Michael Poyer's lab. He's been the key driving force for getting this all working. But we basically, reconstitute the nucleosome sample, tetranucleosome sample in the Poyer lab. Our lab makes the DNA origami, or say Calvin does this in our lab, the DNA origami. We have these biotin labels at the end here. Then the tetranucleosome gets labeled with nutravidin on both ends. Everything gets purified. Then we mix these guys together and are able to get this tetranucleosome sample nicely incorporated between the hinge arms. What we often see is the arms and these kind of two dots that are indicative of this compacted state of this tetranucleosome sample here. And so Calvin goes and does lots and lots and lots of TM imaging, right? We measure end-to-end distances here, again, get probability distributions for that. We do that for the free hinge. And then we also do that for this nanocaliper with the tetranucleosome sample incorporated. And again, using Boltzmann inversion, we can turn those into free energy landscapes. So we get the free energy landscape for the caliper with the tetranucleosome sample and the caliper by itself. And if we subtract those guys, right, that can give us the free energy landscape of just the tetranucleosome sample. Right, so we're able to reconstruct part of this free energy landscape of the tetranucleosome sample with this approach. And again, now we take advantage of the fact that we have different devices that can apply different forces and basically probe different end-to-end distances. And so we individually get free energy landscapes for these different caliper devices. And then we can stitch those together to get this free energy landscape that you see here in the circles for the tetranucleosome sample, basically covering a range from very short end-to-end distance of a few nanometers up to almost 100 nanometer end-to-end distance. And we were happy to team up with Bin Zhang's lab at MIT. They have this nice coarse grain model of tetranucleosomes, and we see that our experimental measurements actually agree quite well, especially at these larger end-to-end distances with their predictions of the free energy landscape of this tetranucleosome subunit. Of course, we notice that at very short end-to-end distances, our model predicts a plateau where their model predicts another free energy increase. But we know that in their model, that doesn't really capture very well the effects of these divalent salts. So if we move to lower divalent salt to 0.25 millimolar magnesium, we see that we do actually capture this increase in free energy at short end-to-end distances. And so this gave us some confidence that we really, measuring something really indicative of the tetranucleosome free energy landscape. And I should stress here that there's no fit here. This is purely a prediction of the simulation. And so we were pretty excited with the agreement here. And the other, key advantage of this is not only can we just measure the free energy landscape, we can actually correlate that to structure. Right? And so we often see this compacted state of the tetranucleosome that we call this two dot or two nucleosome stacking interaction configuration. But we do often as well see this three dot state where one of the stacks has fallen apart, and even this four dot state where both stacks have come apart and you just have four individual nucleosomes. So we have enough data on the two dot and three dot to get basically decouple those and get free energy landscapes for these individual states for the fully compacted tetranucleosome and the partially compacted tetranucleosome. And so this allows us to basically see, where are there biases toward where would it prefer to be in the compacted state, the two dot state, versus where maybe at the larger end-to-end distances, the tetranucleosome can freely switch between the fully compacted and partially compacted states. Right? And then the other thing we can do with this approach is add other proteins that interact with chromatin. So in the interest of time, I won't say too much about this, but we have tested this with this H1 linker histone. And as expected, the linker histone is that this H1 is important in driving compaction. And what we identify here is that this H1 actually introduces about a 3 kBT free energy well at these low end-to-end distances, which is consistent with what other folks have measured. And so this is great, right? We have this nice tool and there's a lot we can do in vitro, but one of the long-term goals of our lab is can we actually get these devices in vivo, meaning not in an animal model, but in a cell. And so because of that, we've had this interest in, how do we start moving towards delivering these devices into cells, in particular into live cell nuclei. Because over the long term, we'd love to have these types of devices inside of a live cell nuclei and use that to monitor and manipulate chromatin directly in live cells. We're not there yet, but that's the goal of where we're going over the longer term. Our lab and other labs also has an interest in gene delivery, right? That's another motivation for getting these devices into the nucleus. I won't talk much about that, but I'll refer you to a recent review paper, if you have an interest to see some of the work going on in that space. But I will share is some of our work really focused on how do we get these devices into the nucleus. And so we were inspired by this approach of a collaborators lab, Laszlo Tora, where they developed an approach to do, we call this piggybacking, right? Where basically they did this with individual antibodies, but they have an antibody that targets a cytoplasmic protein, in this case, an RNA polymerase POL2. And then that antibody gets carried into the nucleus along with that POL2. And eventually, they have an interest in using this to image fluorophore labeled antibodies to image POL2 dynamics inside the nucleus. Our idea was, can we just add a little bit larger cargo, the DNA origami, onto that antibody and see if that same interaction can then carry the origami into the nucleus. And this is all the work of a talented postdoc, Goldberg, who was co-mentored by myself and Michael Poirier. Again, I won't go into the details, but Goldberg developed this nice approach to functionalize origami with antibodies or multiple antibodies. For a long time, we tried this with full-size origami that tend to be quite large, about 5 megadalton, and we didn't have much luck. Every once in a while, we'd see one fluorescent dot in the nucleus that suggested, okay, maybe we're able to get something. It's not too surprising. These origami structures, typical origami structures, are huge compared to an antibody. So we decided just to make a smaller origami. So we cut back to a structure that's about 0.5 megadaltons, label that with 1 or 2 antibodies. We can purify that to get nice pure populations of just the origami by itself, or with 1 or 2 antibodies here. And then
just to let you know, about 4 more minutes or so, approximately. Yeah.
Okay, sounds good. Yeah, thanks, Sonia. I have just a few minutes left. Thanks. And lo and behold, with this smaller origami, we now start to see that we're able to get origami inside the nucleus. This imaging that was done in Malika Lakademiali's lab, but we're able to visualize that with 1 antibody or 2 antibodies, we get significantly more origami showing up inside the nucleus. I won't show the data here, but we also verified stability. We have actually structures labeled with 2 fluorophores, and we see that those 2 fluorophores stay co-localized up to 24 hours inside the cell. So it seems like the origami can stay stable inside the cell for up to 24 hours. What I kind of think is funner to look at and more convincing even is these live cell imaging. And so here, what you'll see, I'll play the videos in a second, but what you'll see here, these are U2OS cells with the nucleus. The blue is labeled the nucleus. And here we have on the left structures labeled with this POL2 antibody. Structures labeled with this maltose binding protein antibody. That's just an off-target antibody, nothing that should be in the cell. The origami by itself, or just the control, nothing added to the cells. Here you see the live cell images. You see all these dots in the nucleus are indicating the origami. But in the video here, you nicely see all these origami moving around inside the nucleus, only in the case where we have them labeled with this anti-POL2 antibody. And so we're excited that we can get these structures into the nucleus. Where we're going now is better understanding what are the effects of size and shape on the cell delivery and nuclear entry and transport, and how they behave inside the nucleus. Can we explore other targeting mechanisms? Say if we want to target a certain region of the genome, maybe use a DCAS9 targeting. And of course, we'd like to get these reconfigurable DNA nanodevices inside of cells and inside the nucleus. And just very briefly, I'll show you something else, recent direction we're excited about. I mentioned we're interested in delivering these devices to different locations of the nucleus. One of our recent interests is also basically delivering or installing DNA origami devices in the extracellular matrix. So binding them to collagen fibers and using them there to measure, for example, shear flows or other kinds of forces, or maybe using them as ways to detect cytokines that are transporting through the extracellular matrix. And these are pretty recent results that we have actually developed this approach to functionalize collagen using these anti-collagen, anti-type 1 collagen antibodies, and get these little origami devices, these hinges in this case, to attach to collagen fibers, shown here by TM and by fluorescence imaging. So this is another direction that we're excited about and moving in the future here. And so with that, I'll end. I tried to mention the collaborators and students and postdocs throughout. Most of this work was funded by these 2 NSF grants. And yeah, I'll stop there and happy to take questions. And just the last quick thing I wanted to share You may recognize this person who I think is actually on the organizing committee. This is a picture of myself and Andre, Professor Kashmirul at Princeton. So I would just maybe a note to the students to do stuff outside the lab as well. Yeah. So yeah, I'll stop there and happy to take any questions.